Okay, so I want to go over this medical press release labeled Scientists Test a Bispecific Antibody that helps T cells zero in on treatment-resistant cancers. So, there's a lot to unpack here, but I think I'll just jump into it and then I'll look up stuff as explanation needs to be made. So, although immunotherapy has achieved increasing prominence in the panoply of innovative cancer treatments, it remains an imperfect tool. Too many tumors simply do not respond to immunotherapy. So to the rescue is an evolving class of engineered proteins that go by the unusual name of bispecific antibodies. And as their name implies, these proteins have dual recognition capabilities. They are engineered to home in on a T cell surface receptor as well as bind to the surface antigen of cancer cell itself. The aim is to bring the two cells together and to activate the tumor annihilating capabilities of T cells. So you may have heard of T cells before, and so T cells are not the they're not the cells that produce antibodies. They are cells that will look for and recognize peptides that are not supposed to be on the surfaces of cells. So let us just look up what a T cell is explicitly. So they are lymphocytes and they mature in the thyroid, which is why they have the name T cells for the T in thyroid. And so let's look at what exactly they do. So let us go to the types of T cells. So this is what their roles in immune defense are. So for instance, the T helper 1 cells, they produce a particular type of cytokine, and they have a key transcription factor, which is, in this case, T-bet, and it produces an inflammatory response key for defense against intracellular bacteria, viruses, and cancer. And if you have an autoimmune condition with these, you can get multiple sclerosis, type 1 diabetes, the like. So regardless, let us just know that, let's go to the cytotoxic T cells, for instance. So the cytotoxic T cells, which I presume will be the ones that are going to be predominantly targeted by these bifunctional antibodies, they destroy virus-infected cells and tumor cells and are also implicated in transplant rejection. So if you get a transplant of an organ and your body rejects it, that is probably due to the T cells attacking it because it does not recognize the peptides that are on that particular organ. Anyways, these cells are defined by the expression of the CD8 positive or the CD8 protein on the cell surface, and these cells recognize their targets by binding to short peptides. So these are 8 to 11 amino acids, and they are associated with the major histocompatibility complex class 1 molecules. And they are present on the surface of all nucleated cells. So every cell that has a nucleus in your body will have these MHC class 1 molecules, and these MHC class 1 molecules will have a peptide that is on its surface. And where do these peptides come from? These peptides are coming from the inside of the cell. So as the cell is consuming things, or as the cell is producing proteins, some of those proteins get destroyed and then turned into peptides, and then those peptides are put onto the major histocompatibility complex class 1 molecules. And then what happens is this complex then gets presented on the surface of the cell with the peptide attached to it. And this allows T cells to scan or look through the body and scan over the cells, and then if it can come into contact with a major histocompatibility complex class 1 molecule that has a strange or a unique peptide that is not supposed to be there, that could be indicative of, say, a virus infection where virus proteins are being synthesized. That could be indicative of cancer because some cancers are going to be mutating frequently, and thus they might have some very obscure and strange peptides in it, which a T cell can then recognize. Now, unfortunately, some cancers actually do not produce sufficient levels of the major histocompatibility complex class 1 molecules. And so this is one of the ways that they are able to evade the immune system by basically removing its peptide or its antigen presenting capabilities. So, just so you know, after these CD8 proteins bind with the MHC class 1 molecules with the unique peptides or the foreign peptides, This causes the CD8 cell to produce key cytokines such as interleukin-2 and interferon gamma, which influence the effector functions of other cells, in particular the macrophages and the natural killer cells. So just know that these cells, after they bind to the MHC class 1, then they start releasing chemicals that help facilitate in the killing or the programmed death of the cell that it's attached to. And then it also helps recruit other other immune cells to the site of affection. So that's just what the cytotoxic CD8 positive T cells do. And there are other T cells, but I believe that those are probably going to be the ones that are targeted by this bifunctional antibody. So let's continue. So they are, as their name implies, these bispecific antibodies, as their name implies, these proteins have the dual recognition capability. They are engineered to home in on a T cell surface receptor, so a receptor that is on the T cell, as well as bind to the surface antigen of the cancer cell itself. The aim is to bring these two types of cells close together to activate the tumor annihilating capabilities of T cells. So let us look up what a bispecific antibody is, just so that we have a better understanding. Okay, so this, I believe, would be the best image to describe it. So here we have our antibody, and normally an antibody would have the same, I don't know what to call it, epitope? No, not epitope. The same, I should say, highly variable region here and here. So if it was a normal monoclonal antibody, you would have the same, I guess, lock here and the same lock here. And you would only be able to bind to one particular thing. Or you could probably bind two of a particular thing, except you'd have to bind two separate ones, but of the same thing. Regardless, I'm rambling. So normally, your antibody will bind to the antigens, and then the back part of it will bind to the Fc receptors. So these Fc receptors are on a bunch of different immune cells, and they find antibodies, and then when they reach the antibodies, they elicit their particular response. Now, normally, let's say we were doing normal immunotherapy, we would go in and try to find out what types of antigens are being presented on the tumor cell, and then we can probably design an antibody that will attack explicitly that receptor. And that could be beneficial if we want to activate the macrophages, the natural killer cells, or the dendritic cells. So this would work for that, but it would not work for these T cells. It would not work for the T cells because the T cells do not have the Fc receptor. Okay, so what is a better thing that we can do? Well, rather than have two of the same epitope recognition sites, let us genetically engineer this entire thing to have two separate ones. One of them will bind, well, in this case it is the CD3 receptor of a T cell, but that doesn't necessarily have to be the receptor that we're going for. Regardless, this one will then bind to the T cell, and then this other site will bind to the tumor cell. And this would be a way to get your T cells right up next to your tumor cells, and you would hope that the binding to the antibody would be in of itself sufficient to help elicit this pro-killing response. So this basically would make your tumor cells vulnerable to both macrophages, natural killer cells, dendritic cells, and the T cells. So let us continue. So research on the bispecific antibodies is underway at the Regeneron Pharmaceuticals, a leading biotechnology company in Tarrytown, New York. The company has garnered a spotlight for its development of REGN-EB3, which is a triple antibody cocktail, which outperformed other investigational treatments last year for Ebola. The drug is under review by the U.S. Food and Drug Administration and is expected to receive full approval later this year. Regeneron scientists are also conducting research on an antibody-based medication that can prevent or treat COVID-19 depending on the patient's needs. So this company has feelers in many different diseases. Anyways, Regeneron's anti-cancer innovation, meanwhile, has grown out of a sobering reality. Certain cancers have developed deceptive deceptive strategies allowing them to resist immunotherapy. Resistance among cancers is as daunting a concern as infections caused by drug-resistant bacteria. So several common cancers have a noteworthy history of thwarting checkpoint blockade immunotherapy, a treatment that relies on the strength of T cells to kill tumors. The investigational bispecific antibodies are designed to help overcome cancer cell resistance. So, Janelle Waite and Dimitri Skokos are part of a large Regeneron team testing a class of co-stimulatory CD28 bispecific antibodies. So remember, this one, we were talking about the CD3, which is one type of receptor, but here they're working with the CD28 antibody, or CD28 receptor, which is on a subset of T cells. So let's look up what CD28 binds to. So CD28 is one of the proteins expressed on T cells that provide co-stimulatory signals required for T cell activation and survival. So this is one of the receptors that if it gets activated or if it gets bound by a proper agonist or a proper ligand, which in this case will be our antibody, it will help convince the T cell to activate and remain alive. So T cells frequently undergo a process of death, right? So they're multiplying and dying, and you want them to be gone when you don't have an infection or you don't have a tumor, but you want them to be there at the location because these immune cells are damaging. Immune cells there's a lot of collateral damage when it comes to your immune system. So one of the ways that we avoid collateral damage is literally just kill the immune cells when they're not needed. Let them die. Once they're no longer giving the signal to survive, they just die. So this is a really important self-regulatory part of the immune system. So T cell stimulation through CD28 and the addition of the T cell receptor can provide a potent signal for the production of various interleukins. Interleukin 6 in particular, and this is one of the very pro-inflammatory cytokines, which is very beneficial at killing cells. So, just so you know, this is another one of the receptors, and the T cell receptor is one of the main, or actually the main archetypal receptor that is on these T cells that help convince them to kill another cell. So, and that T cell receptor is actually the receptor that helps bind to the major histocompatibility complex class 1 molecules. So, just so we know, this is trying to not only bring the CD28 and by effect by proxy the cell that has the CD28 close to it, it is also activating the cell to be more likely to kill whatever cell it's bound to. So the scientists reported their advance in the science translational medicine. So checkpoint blockade immunotherapy is itself an innovative form of cancer therapy that relies on drugs known as immune checkpoint inhibitors. This class of therapeutics is designed to treat multiple forms of cancer by engaging the body's immune system, its T cells, to recognize and attack malignant cells. Keytruda, a medication that helped revolutionize the treatment of non-small cell lung cancer, is a checkpoint inhibitor. So all checkpoint inhibitors are based on a deceptively simple principle. Cancer cells possess a protein dubbed PDL1. T cells have a surface protein called PD1. Tricky cancer cells use their PDL1 proteins to elude T cells to get past the guards, or the checkpoints, an activity that allows tumors to proliferate and spread. So T cells normally have these PD1 receptors, right? And these PD1 receptors are interacting with the PDL1s in some way let's look this up, because I might as well. So PDL1, so this is programmed death ligand 1, also known as cluster of differentiation 274. So PDL1, is a transmembrane protein that has been speculated to play a major role in suppressing the adaptive arm of the immune system during particular events such as pregnancy, tissue allografts, autoimmune disease, and other diseases such as hepatitis. Normally, the adaptive immune system reacts to antigens that are associated with the immune system activation by exogenous or endogenous danger signals. So normally you have these danger signals that are in your body, and then those danger signals are what are activating the innate immune response. The adaptive immune response of your immune system. So in turn, clonal expansion of antigen-specific CD8 positive T cells and or CD4 positive helper cells is propagated. So normally these antigens will cause the CD8 cells or the CD4 positive T cells to duplicate more so that they can attack the pathogen or attack the cancer, right? The binding of PD-L1 to the inhibitory checkpoint molecule PD-1 transmits an inhibitory signal based on interaction with the phosphatases SHP1 and SHP2 through an immunoreceptor tyrosine base switch motif. Long story short, this PD-L1, which is on some of your cells, is going to inhibit the process of T cell proliferation and activation. So this reduces the proliferation of antigen-specific T cells and lymph nodes, while simultaneously reducing apoptosis in regulatory T cells. And the regulatory T cells, these are going to help suppress the other T cells. So you're basically, with PD-L1, you are stopping the regulatory T cells from dying, and you are preventing the other types of T cells from multiplying. So you're creating an anti-inflammatory response as a result. So that's what PD-L1 does, and a lot of cancers actually upregulate PD-L1, which ends up tricking the immune system. Oh, this isn't something that I need to attack. So anyways, multiple cancers that range from Hodgkin's lymphoma to lung, bladder, ovarian, and kidney cancers may initially respond to checkpoint inhibitors, but soon develop resistance. So basically, the cancer cells mutate and evolve in a way that makes them resilient to these checkpoint inhibitors. Anyways, the Regeneron team studied 2 bispecific antibodies that each target the T cell protein dubbed Cd28. At the same time, they analyzed 2 tumor-specific antigens. The bispecific antibodies attracted both T cells and cancer antigens, enhancing the potential of cancer cell death by T cells. So, Waite and colleagues found that bispecific antibodies enhance the effectiveness of treating the anti-PD-1 checkpoint blockade in mouse models. The scientists also say that combination sensitized previously resistant tumors to treatment. So this is basically saying they found explicitly tumors that were resistant to this particular checkpoint inhibitor. And by being resistant, it made for a prime candidate for this new cancer research. And this new cancer research said, yeah, by using these bifunctional monoclonal antibodies, we are now having the benefit of, or we are now sensitizing those new tumors or those resilient tumors to normal immunotherapy. So this is great. So monoclonal antibodies that block the programmed cell death checkpoint have revolutionized cancer immunotherapy. However, many major tumor types remain unresponsive to anti-PD-1 therapy, and even among responsive tumor types, most of the patients do not develop durable anti-tumor activity. So in a series of animal studies, Waite and colleagues demonstrated that the experimental class of dual affinity antibodies can safely boost the cancer-killing power of checkpoint blockade immunotherapy in mice. The animals had tumors that tend to resist immunotherapy treatment. The results represent a stride in creating safer cancer immunotherapy combinations, the team reported. In addition to the mice, the antibodies were additionally well tolerated by long-tailed macaques and did not cause the severe immune side effects that have held back similar treatments in the past. So this is amazing that in a primate model, we do a lot of our FDA trials and experiments on these primate models. And a lot of times if there's complications that arise from these treatments, we can see it in the animals first before we bring it on to humans. Now, one of the downsides with this particular type of this field was that we saw a lot of problems with the macaques, and that's very problematic, right? But this particular bifunctional immunotherapy seems to be able to, or it doesn't have those severe immune side effects. So the research arrives amid worldwide efforts to address the obstacle of tumor cell resistance to checkpoint inhibition. Last year, scientists in France advanced the notion that rotavirus vaccines can be used to overcome the problem of cancer cell resistance to checkpoint blockade immunotherapy. That is interesting. I might need to read up on that later. So we have found that rotavirus vaccines Rotatex and Rotanix have both immunostimulatory and oncolytic properties. So oncolytic, the ability to destroy cancer. So Dr. Tala Shinkarian of the Center of Research in Cancer of Lyon told Medical Express. She added that vaccines can directly kill cancer cells with features of immunogenic cell death. So Shikarian, Lightweight, and colleagues found a way to overcome tumor cell resistance. The French team additionally underscored the importance of having an inexpensive off-the-shelf solution to a stubborn problem in cancer care. So this is good because so vaccines often are immunostimulatory in one way or another. So it's really nice to see that there are some other ways that we can elicit similar responses that does not necessarily necessitate monoclonal antibodies. So just so you know, antibody therapy is one of the most expensive, or it's a pretty expensive therapy because you have to produce these antibodies in some sort of system and then purify them and then sell them. And proteins are very, antibodies are large molecules, right? They're very large, and because they're very large, there's just more room for error. And so this entire process is probably extremely expensive. So it would be nice. It is nice when you have a cheaper alternative, such as a vaccine, right? So with that, I think I'm going to leave this. But yeah, I find this very interesting, and hopefully that there is more that will come out of this. So with that, thanks for watching, and hopefully I will see you tomorrow.